Hello and welcome everyone to CyVerse's Focus Forum webinar series. I'm Tina Lee, CyVerse's Engagement Officer, User Engagement Officer. It'd help if I knew my own title, but I never use it. Anyway, today's webinar is on how to scan and visualize genomes using PatMatch and Circos in the CyVerse discovery environment, presented by Wayne Decatur of Upstate Medical University in New York. For those unfamiliar with CyVerse, we're an NSF-funded cyberinfrastructure project, and this free webinar series helps fulfill part of our mission to train you, the next generation of scientists, on how to use CyVerse's computational resources. So we're going to take care of some housekeeping really quickly, and then we'll head on to the webinar. Today's presentation is about 30 minutes with time for Q&A at the very end. Please open your Zoom chat window and type any questions you have there for Wayne. Materials from today's webinar will be posted on a wiki page for which we'll email you the link later today, and that'll include things like the presentation slides and the video. Wayne also has a URL where you can find all this information as well. I will launch a poll shortly for your feedback on possible topics for our fall webinar series, so please be sure to vote. And if none of the topics interest you, by all means, send me your suggestions in the chat window or email me. We should have our fall lineup set by later this summer, and we'll post that on our CyVerse website. Also, in just 2 short weeks, CyVerse will start its inaugural Foundational Open Science Skills training, a camp-style event from June 3rd through 7th here at the university. We're aiming that at early career researchers who want a solid foundation in open source resources for doing open science. So visit our website for more information. Okay, without further ado, please meet Wayne Decatur, a researcher at Upstate Medical University's Department of Biochemistry and Molecular Biology. Hello, Wayne.
Hi, thanks, Tina. It's a pleasure to be here and get to speak to everybody watching about PatMatch and Circos and running them in your browser, which hopefully will make things much easier for everybody. First I'd like to start out with an outline of what I'll talk about today. First I'm gonna go like a big picture perspective, because I think it's helpful to have a context of where and how you're running these tools. At least some idea. And then I'm going to go into PatMatch and do a demo of that. Then we'll come back and we'll talk about the benefits of using the CyVerse Vice. And in case I forget to say it, that's the visual interactive computing environment of that CyVerse has recently introduced. And then I'll actually introduce Circos and running a demo in there. And as Tina alluded to, I've made because there's a lot of links and things, in order to make it much easier on everybody, right up front, I'm putting this link here that this is all you need to record. And when you go to it, it'll take you to a page that looks kind of like this at the top. I have to apologize that you do have to be careful of the case, so the C is capitalized and the W. But that basically parallels what I'm going to talk about today. And then CyVerse will provide you with the additional information and links to the video later. So as we talked about, I'm ostensibly here to talk to you about PatMatch and Circos, and I think these actually bookend very well, because PatMatch is kind of straightforward. By the end of this, you'll probably be an advanced user of PatMatch, whereas Circos is actually, because it's actually a lot of artistic and creative features to it, there's no way I'm going to be able to touch on it, and I think people probably just be disappointed that we're just touching on some of it, but I think it'll give you a good foundation, and you'll see that with these resources, you can actually run both of these very easily, and that way you can become useful. And in fact, a lot of these other command line tools could be done this way too, and we'll touch on that at the end. So I want to start out with this diagram by Juliet Taka, which kind of gives the overview of what's the golden standard that we're trying to approach in publishing of science. And you can see up in this, she has a paper. You can imagine maybe it's just a paper that has some computational stuff, not all of it has to be computation. It can be experiments and computation. But what would be nice is if anybody could actually run that just as she had run it. And a lot of people want this to become the standard. And the idea is that we actually have the computer resources now to do a lot of that, to actually give everybody the exact environment that people are using in addition to the code and everything, instead of just list of versions and software. So I'd like to expand on this if you'd oblige me. So I'm going to adapt this, and I think a lot of this you don't need to know what's going on behind the scenes, but CyVerse fits very well along with what we're going to use today. In addition, this binder system to kind of run this kind of thing. And in fact, that's what this original diagram was made to highlight the binder, but I'll say that CyVerse fits in right there. And I'd also, if you note, I also changed up here, instead of saying notebooks, because those are kind of sacred to experimental biologists, and we're not talking about electronic notebooks. We're talking about something very special. I think it's better to think about these as digital. You're providing a digital workbench where it's just like your experimental workbench. You have all the same equipment, all the same disposables, the pipettes, everything is the same. So I think workbench fits, and digital workbench describes this kind of environment that is provided much better. But let's rewind time a little here. Imagine and get back to closer to what we'll be doing today. Is you probably have data and you want to use PatMatch or Circos to analyze them. And so that fits in better here. And you can start to think of it as it's going to be an iterative thing, because you're going to get input from people and it would be nice to share with them. Maybe not the public yet, but your collaborators. And in fact, one of the things a lot of this is based on helping you is future you, or help the past you make it easier for the future you. In fact, most of you probably collaborate the most with the present and past versions of yourself. And so that's what I want to highlight in addition to this. So let's again rewind a little and talk about what we're going to do today. So we're going to tap into this to be able to use PatMatch and Circos, and you'll see how that fits in. And one of the ways of doing that is if you start building this in early enough, when you've got to go and you want to put it together with a publication, it's going to be much easier if you've been doing this all along, and it will make it much more reproducible. One thing I'd like to point out is that this isn't niche software or anything. This is the Jupyter system, and which also allows RStudio to be run. This is world-class open software. In fact, recently the Nobel Prize in Physics went to the LIGO and Virgo groups, and you might have seen the pictures of the black hole, of which actually some of that stuff was run on the CyVerse system itself. And I just want to say it's not even limited to academic software. Netflix relies on Jupyter a lot. Okay, so let's go back and talk about PatMatch, this tool. So what does it address? It helps you find sequences or patterns in nucleic acids and proteins. But you might ask, why is it better than other tools that I can use? One thing is you don't want to open big, huge files of sequence files in like your normal graphical user interface and try to use find and replace. It's just going to be a nightmare for yourself. You got to think about line breaks. And so that's a lot of the stuff that PatMatch brings to the game here. And also this big thing I want to point out is it provides biological syntax. And we'll definitely see it enhances scalability and reproducibility. So, but to the biological syntax, I'd like to point out this, is that you can do ambiguous or different amino acids and proteins and describe them differently. This is just part of the listing of the supported syntax. And the place I like to refer to it is the yeast genome, actually, which is where PatMatch started, and then it moved to the Arabidopsis. So there's actually web servers that you can plug into to just search individual genomes, but we're going to see today, this allows you to search any sequence. And by doing it on command line tools, it'll be much more reproducible, even if you are doing some of the model genomes that are out there. And this is just the rest of that thing. You can see that you can do complex patterns where you allow certain residues exactly 16 times and things like that, different, and you can mix and match. Okay, so I'm going to do a demonstration. It's not actually going to be completely live. I pre-recorded it just a few minutes ago, but for the sake of what you'll see, you could actually be running it right in parallel with me using that link I gave you earlier, and it'll run. So we're going to run the PatMatch, and we're going to use the binder version this time. We're going to come back and we'll talk this way. It'll kind of set you up. You can see the difference between binder and CyVerse Vice apps as we go along. So here's the page you want to go to start running this. The link's up at the top. Let me start this. There we go. You'll see, and then you want to click this Launch Binder down at the bottom there. And as we click Launch Binder, you see you didn't have to sign in or anything. It's completely anonymous. And then, in fact, that's part of that's why there's some restrictions. You can click that Show to see how it's running. And it usually boots up pretty fast. But this is what you'll always see, because really what it's doing is spinning up a system. Now, the first notebook, I'm going to say skip that. That's kind of for people who already know about PatMatch and just want to use it on the command line. And you quickly get ushered to the second notebook with PatMatch with Python basics. And this steps through really how to use it and use it in conjunction with Jupyter and Python. So first, we're going to get some sequence data by running that first cell, and you just hit Shift Enter to run cells, and then we'll see whether we got that sequence. It's listed in the directory, and then what you have to do is deal with making it all one line. So that's what this. So PatMatch comes with this script that just does that. So you always have to run that. You get your data and you run this. And then now we're ready to run PatMatch. So we just go down here and we click on this and we give it a pattern and we say we want to use that script. And you see it's just boring text. It's got all our data there, but it's a little dull. And we can see the rest of this is actually going to be showing how combining it with the Jupyter system, you can quickly get to stuff that's a little more fancier and a little more informative. So what we're going to do here is we're going to save it as a file instead of just that text we saw to a screen by running that cell. And then we're going to get a script that's going to allow us to bring it into the data frame, which is you might have heard before, it's a way to handle large amounts of data, kind of like a spreadsheet, but much better and much more powerful. So we're going to go down and run this one where we tag it with we're going to give it some additional options that help make things a little clearer later. And now we're going to see that we did that. We're going to actually bring it in because it saved it as a file. For now, we're doing it the kind of long way. And when we run this, we see the data frame and we get this nice format, much more clear than that text space that we had before. And then I'm scrolling down and we can use that data frame to do some other things such as plotting. So right now I'm going to run this line and we're going to import a script and we're going to bring that in. And then we're going to change some column names so that it works. And then we're going to run some plotting. And you can see the promoters plotted across the genome. And we can do that with a bunch of things just by adding a few more lines. And we got all these restriction sites and things. And you can see the palindromic nature because each of the restriction sites is double there. And then it just continues on. It shows you how to bring it to Excel if you wanted to do that. And then back on the index, what it eventually gradually leads you to, because I did this for people who maybe don't know about Jupyter or Python, because PatMatch is actually originally written in Perl. Well, it actually still is written in Perl. So you're actually running Perl stuff. But the advanced notebook takes you through really how you would actually run it in current environments without using all those file intermediates that we saw it making in the original one, which makes it a lot more streamlined. Okay, so that's the demo of PatMatch. So let's go and I want to show the difference between CyVerse and Vice a little before we get to it, but talk about it. So both of them give you the installation, no installation is necessary, which is great because you get portability. It's not going to impact your local system. You don't have to worry you're clobbering what you already have working. A lot of people are seeing this, especially because Python is converting over to be purely Python 3. There's no more Python 2.7 soon. So with these, you get both of that, plus you get a lot more, but that lowers the barrier. And here's what else you get with both of these. But I'm not going to go through all the lists because I think some of it you have to experience it yourself and start to understand what you're getting. But I do want to point out with CyVerse Vice, you get some bonuses here in that you get better connectability in shareability. We can talk about what that means in particular later, but at least the shareability means you can decide who you want to share it to without making all this public, whereas the binder system has to be public. And you also get better persistence because you can actually run the CyVerse version of this Vice app for 2 days without any problem. Whereas the binder one, you're limited to it only 6 hours, and you have to keep it active. If people have questions about that, we can talk more about that in the questions as well. Okay, so now let's finally get to Circos. It generates informative visualizations, but that's a picture here will be a lot more informative. So what I want to do is go through. You can find this right at the Circos main site. This is kind of really shows everything it can do, and I'm going to kind of zoom into some of these features. Because up in the corner is really kind of the most advanced we're going to get. In fact, we won't even get that advanced unless it comes up in questions. But what it can do is all you have to do is give it a summarized details of the chromosomes, and it'll draw them out in this circular fashion. And we'll get back to why that might be helpful soon. But I want to zoom in on this part here. And so one of the things so some of the things you get besides just the chromosomes that we just saw so there's the chromosomes. You can also add data tracks. You can also add what are called links. And the links allow you to show relationships. For example, here we can very clearly see the middle of dog chromosome 1 is actually highly related, meaning that the neighborhood of all the genes that are encoded on it are very similar to human chromosome 5, and over here in the yellow is 9. So to contrast that, I'm going to show this, which is the typical linear plot. But if we wanted to do the reverse now, if we wanted to look at human chromosome 5 and 9 and see how they relate to this different organism, Amphioxus, you can see it's much more difficult. And I'm not trying to say anything bad about this picture because this really gets the point across of what it needs to get across. But you can see if you were interested in certain neighborhoods of chromosomes, you would want something more like what Circos can make. So so what do you need to do a Circos plot? The minimum you need is a karyotype and an ideogram. And these are actually defined in the configuration file. And they kind of parallel what people are, if you're used to do karyotyping, and ideogram is just another word for plotting. But so the karyotype has the name and the size of the chromosomes and the cytogenic banding, if it's available. And the ideogram just tells you how to plot that, basically. And why this is minimum, because the boilerplate can basically take care of all the other information. But on top of that, you will usually want to enhance it with, you usually have links that we saw that can show relationships, and data tracks, and ticks and labels, and different highlights. And that we saw it all illustrated in that previous example. So that's kind of what you're working for. And that's actually the best way about making a Circos plot. If you kind of have something similar to what you want to make using your data, you can usually, it's best to start with the code example of that if you can find it. And I give some tips in the expanded, in that link, in the expanded version of this about where you can find some of this. But we'll see that we have a lot of resources right in this, right at CyVerse Vice app that we're going to run here. But one thing I should point out is, so the karyotype and the ideogram, all those are very summarized. Well, the karyotype is definitely summarized data, so you're not inputting a lot of data there, but your data tracks and your highlights can start to be a large sources of big data, and that can kind of be memory intensive. So you have to watch out that, and that's one place that CyVerse is going to be better than the binder system, because you're a little more limited in the binder system. So if you suddenly see it stopping in the binder system, you're going to want to be using CyVerse. So this is kind of a segue to actually running the demo to better understand what's going on. So what you need to run Circos is actually you just point the software at a configuration file, and the configuration file then is telling Circos everything else it needs to make the plot. So the minimum configuration, I'm going to go to the next slide, and we'll see, we're going to expand this to see the full thing. And really what it is just showing the points of the karyotype. You have the ideogram there, and then again, the rest is handled by boilerplate if you want to. So that's all you actually need to run it, and that's what we're going to do in the demo. But to understand how the demo is working and how you can go beyond that, I want to show you kind of what is there. So if you go to the Circos site, there's this whole tutorial system, and there's different sections, and each one has a different page that illustrates one aspect. And in fact, this goes way down. There's like 10 sections. I'm not showing all of this. This is just the top of the page that you kind of see. And in fact, it's an edited version of that. So, but once you go into the Vice app or the binder, what you'll see is in the tutorials, which we'll now click on. So this is the tutorials directory that'll be available to you when you access that. What you want to do is basically what we're doing is we want to drill down, find the configuration file associated with each tutorial, and point Circos at that. And that's really all it's necessary to run all the tutorial examples. So now we go in and say we want to run section 7, and then there's there's actually 8 sections of section 7. So there's 8 pages, and say we want to do page 2, we would point it at this, and finally we would get this. What we need is this, what we're looking for is the configuration file, and that's what you want to point everything at. And then the configuration file handles pointing everything else out. And in fact, this is what you're going to generate this image. Because the tutorials are already set up, they already have the images generated, but for the sake of the, I hemmed and hawed whether I was going to delete all the images to make sure that you're actually showing them, but I left them in for the sake of this, but we'll actually run it. So we're going to do a live demo, and we're going to actually step through the Hello World style version of this. So you need to go to the Circos binder page. There's other ways to do it, and we're going to scroll down because we're going to use, let's start the demo here. We're going to scroll down and use CyVerse this time and not the binder version. So if we scroll down on the page, which this is actually the page, you'll see that there's several ways. So if we knew what we're doing, we could just press that launch Vice. But I want to point out that there's steps to guide you when you're first learning it. And we can do it from this page. We can launch Vice. And what you might see at the time I am running this, I'm already signed in, so I didn't need to sign in, but you might need to. And it'll bring up the discovery environment. With that app already opened, you can put in your information. And what I'd like to illustrate is one way. So CyVerse is really nice about being able to connect it to the data. So after I input the analysis name, I'm going to go down here to parameters, and I'm going to press on that, and we're going to open that up, and I'm going to point it at a folder that I already have uploaded to get some data. So I'm going to pick this archive, and I'm going to say OK. And now I'm going to actually add some files from that. In fact, it turns out there's only one, so I'm going to click on that and add that. Okay. And some other people have shown other ways in earlier of these Friday seminars that I've linked to on that outline. And you can see other ways to actually get your data into the Vice apps. So now I'm going to click what I've already loaded with a demo. In fact, it'll look a little different for you, and we have to apologize. I'll get back to this in a minute, but you want to click on the index, and then this index page has parallels that the tutorials I showed you, and then we're going to click on this first one, this Hello World, and we're going to bring it up, And it kind of tells you a little bit about using Jupyter because this is one of the first pages. And you can see that we see the conference file. If we scroll down, we're going to see where we are working in the system. And now we want to change our directory, like I showed you in advance, so we can point right at the Circos file. And we see there's the configuration file. Now we can point Circos right at it using this cell. And that's all we're going to do. Once we run that cell, it's actually going to run Circos on that configuration file. And we can see that now when this finishes, which the asterisks will go away, we'll see that we have this file now, and we can open we can actually, because the greatness of Jupyter, we can look right at the file in here. And if we wanted to, we could look at SVG. I don't do that in most of them because it's a little demanding of, the files start to get large, which isn't a problem in CyVerse, but in the binder system, it might be a problem. And you can see we've got a host of other tutorials. Depending on the timing and how many questions we have, if people want to, I could show some of the other ones. But as I said, you might be a little disappointed. We're not gonna run a large demo on Circos. But there's all these ones will run just as tutorials. Except I should point out, right now it won't work in the one launch from CyVerse. There was a minor glitch that we were trying to work out remotely because Apendra, who I'll refer to in a minute, he's actually was away, and so we were trying to do this not being on the computer at the same time, and it just didn't work out. The PatMatch system, though, will look just like everything I showed you, whether you use Binder or CyVerse. But in a few days, the CyVerse version of Circos will look like this, so I have to apologize right now. But it actually run. If you know what you're doing, you can run it. So I should point out that thank the software people that actually made what we're using. So Martin Karinsky, he did Circos and made this amazing site where you can learn more about it, and hopefully using CyVerse or Binder, it'll make it a lot easier for you to learn about that. You can do it right in your browser, not have to install anything. And then this is the list of people who wrote PatMatch, and I have to thank the CyVerse team for letting me speak to you today and giving me this great place to run this. And thank the Binder team, who allows a very similar kind of tools to public data. And I have to thank my department and my PI, Mark Schmidt, here at Upstate for supporting me. So if you have suggestions or needs for Vice apps, I don't know if Apendra knows this, but I'm throwing his name out there. If he's not the person to talk to, he'll make sure you get to the right person. And one of the best ways to contact people at CyVerse is actually use the CyVerse intercom. It's down in the corner when you're logged into CyVerse anytime, and they're very helpful. And I'd just like to reiterate, this is where you can find links to everything I've shown today. And again, the CyVerse people send out more information, such as the video and links to all this. And if you want to contact me, here's my Twitter and my GitHub and my email. If you have any needs for Circos or PatMatch or looking for some help on how to get started in some of this stuff. And with that, I'll leave it at what Tina just touched on earlier, that CyVerse is supported by the National Science Foundation. I'd be happy to take questions right now.
Thank you, Wayne. I don't see any questions typed into the chat, so we'll give a few more seconds.
We covered any. Actually, Shelley had a good one earlier.
Yeah.
I'll use it. When we were practicing, she asked me, what is the limitation on each of these? What size genomes can you use? So I can touch on that. So I kind of touched on it with Circos, is that there really isn't a limit because it's summarized data. It's actually the additional data that you add is going to be limiting. But for PatMatch, I can safely report CyVerse can handle the largest current genome, which is the sugar pine, which is 30 billion nucleotides, the largest assembly I know of. There might be one out there that's since been published, but at least as of 2017, that was the current largest one. And it handled it like a breeze. In fact, that's a link on there if you want to run that on CyVerse. It's under the additional technical things, and you can see it run. So So PatMatch is not limited in running on CyVerse, and it can handle huge genomes.
Okay, a couple of questions have come in. Can Circos be used to compare bacterial genomes?
Yes, in fact, there's a recipe. If you go right there for the first recipe, I forget which section, if it's 8 or 9 or 7, there's some recipes, and one of the examples they show is using it for a microbial genome. So okay. I would start there.
Okay, and there's a follow-up to that saying, can you compare, for example, cow genome and the incomplete deer genome?
So the cow is available. In fact, that's one of the links I use. So that's, the karyotype could already be easily made. That's one thing I didn't get to point out. I made a script that makes it much easier to pull things out from the UCSC genome database if it's already there, which I know cow is. I don't know if deer is there, so you would probably have to make the karyotype yourself, but it should be fully able to do that if you have that. But you're going to have to figure out the relationships. So Circos is purely plotting. You have to actually input the data, and there's a lot of other things that'll do the synteny and generate that data that you'll need to do that.
Okay, thank you. Another question is, what is the format for adding data tracks?
Oh, that's a good question. I've recently reviewed links, which are very easy. In fact, it's just the coordinates. So data tracks, there's actually, thinking about it more, there's actually like 4 different kind of data tracks, and all the formats vary because you can have scatter plots, you can have bars and histograms, but they're all spelled out, and there's examples in all the tutorials show examples of each of those, and I think that's under like section 5 or 6, the data tracks, or maybe even earlier. So I would start there. If we had more time, we could skip through and find that, but I'll try to keep it a little more concise and not wander off.
Okay. Another question is, the Circos ideogram is quite cute for the human genome, but will need to be generated for other fungal and or plant genomes, right? Do you have any suggestions on how to best do that?
Yeah, so anything that's already at UCSC, like I said, you can actually, I've got a script that will make it much easier to do the karyotype than doing it by hand. So you really only need the chromosome size and everything, and they're easy to generate. In fact, you'll see a lot of people when they publish these genomes, they'll generate images. But because they can get rather large, if you want the detailed ones, I don't think there's, and maybe I'm wrong, but they're actually, I think I am wrong. There's some web tools that have consolidated some of these. In fact, the microbial ones, I should point out, somebody did ask a question about microbial ones earlier. So there is a site that you can go to that a lot of the microbial genomes that are already published, you can actually, and it's under the expanded ones. That's not for us. It's under the expanded sheet. There's the microbial ones. I don't know if there is a database that's running this on everything as it's published. Again, because there's limitations on the sizes. As you can imagine, the data gets kind of large with eukaryotic genomes. So if I come across something, I'll probably put it in those expanded links that I have of where you can find information and alternatives. So that's where you can find that microbial listing is under web tools. Okay.
Another question is, can I use PatMatch for apples, or is it strongly linked to the yeast database?
No, the yeast, my yeast example was purely just an example. I happen to work on yeast right now, so I just used what I was most familiar with. If you have any sequence data, you can use PatMatch. You just need to get the sequence files, and so by pointing at the yeast genome database, that was one easy way to get the yeast information and bring it in that example. But I don't know if there is an Apple one, but if you have the Apple sequence files, you can just get those or even drag them in. So the Jupyter Lab version, which was the second version of the notebook kind of interface that we saw, which I was running in CyVerse. In fact, in that, if I scroll back, I'll actually do that. And this, something that looks like this, So in the Jupyter Lab interface, you can actually just drag and drop right into this, what's called the file browser. This isn't available in the older interface that I showed in the binder, which actually is a default, but you can get to the Jupyter Lab if you know what you're doing. So that's always available, and you can just drag and drop even the sequence, so you don't need to actually use any fancy way to get the data in. But yeah, as long as you have the sequence file, you're golden with PatMatch.
Okay, that's it for questions. I'll give a couple more seconds, but basically, thank you everyone. This is the final webinar of our spring series, and as I mentioned earlier, we are working on our fall lineup, so come visit our website frequently to see what we've got. We'll be tightening up those, and they'll start probably in early September. Oh, one more question. I'm just beginning to learn bioinformatics. What's the importance of Circo? Why should we learn to use it?
So Circos is like the premier command line based version of making a circular plots. So if you want to make any of those plots, it's useful. Again, anytime you're going to use the command line, it's going to be more reproducible than going to use a web tool where you can plug in things. But if ultimately your goal is just to get started, those web tools are fine. And in fact, you can make great images with those. So Circos or the related tools are definitely good to go. You'll see people using them, and then you'll want to make some yourself.
They're beautiful, I have to say. So, okay, well, that's it. Let's see. Oh, a follow-up question on Circos. Is this for Synteny plus visualization or only for visualization?
No, see, it doesn't make it won't extract the Synteny information itself. You're just giving it summarized information. So that's definitely an important thing to make. You actually have to bring it in, and that comes from separate places, which I've done some Synteny work, and I didn't generate the output that would go into Circos. So I'll be actually updating some of my own information. So at that time, maybe I'll try to find a way to connect the Synteny to Circos a little better, and I'll update this information. So if the person has a direct question, they can contact me, and I can help them, guide them to working on that a little better. But it doesn't. It's for the visualizing the Synteny, not extracting it.
Okay. All right, any final questions? If not, Wayne, thank you so very much, and thank you all for attending today's webinar. Be sure to vote in the poll if you haven't yet, and everyone, we'll see you come fall. Thanks so much. Bye now.